Welcome student on another video lecture. The topic is biosynthetic pathway or the subtopic which is under the biosynthetic pathway, that is biogenetic investigation. So what we have seen in there are different types of biosynthetic pathways by which the plant will produce primary metabolites, and from that primary metabolites different type of secondary metabolites are produced. But in which part of the plant that particular secondary metabolite is produced or synthesized, that should be investigated, or so that that part of the plant is used as a crude drug for further studies or for medicinal purpose or therapeutic use. For that reason, we must know which part of the plant is going to biosynthesize that particular secondary metabolites, and for that there are various intermediate steps which are involved in biosynthetic pathway that can be investigated by following techniques. First one is use of isolated organ grafting method. Third one is use of mutant strain. Fourth is tracer technique, and next fifth one is enzymatic studies. So all these are the different techniques by which the biogenesis of the plant, where that particular secondary metabolite is synthesized, in which part of the plant, that is investigated. So to investigate that biogenesis or synthesis of secondary metabolite, radioactive tracers are used, and the first one is isolated organ, tissue or cell. The cultures of organ, tissue and cell growing under controlled aseptic condition can be used for feeding experiment. So here the tissue culture or organ culture or cell culture is prepared in a culture medium. The cells are grown, and those cells which are properly grown, a callus will form, and from that callus the cells are transplanted in another medium so that they will grow properly. This is the basic plant tissue culture technique, and in that different cultures of organs and tissues and different cells are done, and the whole operation is kept under aseptic conditions. Strict aseptic conditions are maintained, and after that, when the culture is prepared, that culture can be used for feeding experiment. The radioactive tracers, these are the radioactive isotopes of carbon, hydrogen or nitrogens, they are used, and these tracers are introduced by this process to the parenchymatous tissue of shoot, leaves or root or other parts of the plant. So the parenchymatous tissue or parenchymatous cells, they are selected, and in that cell or in that tissue the radioactive tracer which is labeled, that radioactive tracer is introduced into that parenchymatous tissue or parenchymatous cell. The radioactive tracer, that may be a carbon compound or that nitrogen or hydrogen, and the different isotopes, they are selected and they are introduced into the parenchymatous cell or tissue of shoot, leaves or root or any other plant structure, and that radioactive tracer is now labeled. It is a label compound. Now the further analysis of such plant material, further analysis and investigation is done on such plant material, and which will provide the important information about the incorporation of label compound, and it will determine the site of synthesis of that particular compound. That particular compound, where that particular compound is synthesized, that can be traced by using that radioactive tracer which is introduced into the parenchymatous tissue or parenchymatous cell. So this is the isolated organ, tissue or cell culture technique. In that culture medium, the culture is prepared. After preparing culture, the callus will form and the cells are selected which are healthy and growing cells, and in that cells the parenchymatous tissue or parenchymatous cells of that tissue or shoot, leaves or root, they are selected, and in that radioactive tracers are introduced. These are the label compound, and further analysis on these tracer is done, and that will helpful to determine the synthesis of particular compound. Where that particular secondary metabolite gets synthesized, that will be determined by using that radioactive tracer. Isolated roots are extensively used for the circulation of biogenetic pathways for tropane alkaloids in the root of solanaceous plant. So the isolated roots, they are cultured and they are extensively used for biogenetic pathway of tropane alkaloid, because these tropane alkaloids, they are generally synthesized in the root of solanaceous plants. So isolated roots, they are used to find out the biogenetic pathway for tropane alkaloid. The studies on petal disc have been used for elucidation of pathways for essential oil components such as rose oil. So how the essential oils, they are biosynthesized in the plant, what is the biogenesis, or where the site, what is the site of that formation of essential oil, that can be studied, or the pathway can be elucidated by using petal disc. Petals are selected rose petals, and these petals disc have been studied for the essential oil component such as rose oil. Isolated shoot and leaves can be maintained in a suitable sterile medium for studies on Nicotina and Datura. So here the isolated shoots and leaves, they are selected and they are maintained in a suitable sterile medium for the studies and the of Nicotina and Datura. In such type of studies on rooted leaves, get large organization of root, facilitate the study of tobacco alkaloid. So rooted leaves which get a large organization of root, and which will help to study the tobacco alkaloid for their biogenetic site, and which is generally considered to be the root. So the root of tobacco that contains this alkaloid, that is nicotine, and from that isolated shoot and leaves, the biogenetic pathway can be found out. Next method is grafting method. It is an operation in which two cut surface of different but closely related plant placed so as to unite and further grow together. So grafting is the operation in which two surfaces of different plants, but these plants are closely related. Two plants are selected. These two plants are closely related, and two surfaces or two portions of plants are collected or cut, and these two cut surfaces are then unite. After cutting that surface, they are unite, and after union they are allowed to grow together. A major part of plant which is used for grafting is a stalk. When two plants are selected, one part of plant, that is a major part, and which is a stalk, whereas the portion that cut off from another plant is called scion. So the another plant from which the portion is cut, that is known as scion, and the major portion is the stalk. So the those part of plant which are used as a stalk, the major portion is of stalk, and another portion from plant that is cut and which is placed in such a way, they are closely placed in such a way so that they will unite and they will further grow together. You can see here two different parts of the plant. One is a stalk and another is a scion. These are closely related plant, and two surfaces are cut and they will make closer. Here there are two different surfaces. This one is the major plant and this one is the that is stalk and scion. These will come close together and allowed to grow together. Now they were growing together. So this type of technique is known as grafting. Here the two parts of two different plants is there, but these plants are closely related with one another, and they are placed closely so that they will grow together. In case of plant propagation, grafting has important place, and the plants like cinchona, citrus, myristica, etc. have been successfully grafted for the production of better quality of drug. So in case of plant propagation method, grafting is also one of the method of plant propagation, and in that the plants like cinchona, citrus, myristica, they have been successfully grafted, and after grafting the quality of drug that you will get is a better quality drug is obtained after grafting. Grafting has also considerable utility in the biosynthetic study for elucidation of pathway used for biogenesis of secondary metabolites. So grafting is also helpful in determining the elucidation of pathway or biosynthetic studies. What is the particular part of the plant which shows the synthesis of that secondary metabolite, that is biogenesis of secondary metabolite, and what is the particular pathway that is followed by the plant to synthesize that particular metabolite, that can be studied by using this grafting technique. Solanaceous plant like Nicotina datura have been intensively studied for tobacco alkaloid and propane alkaloid. So solanaceous plant like Nicotina and datura, both they are come under Solanaceae family, and they have been studied for propane and tobacco alkaloid. One of the example of grafting is scion of the tomato grafted onto the stalk of datura. So the major portion is of datura, and in that scion of tomato is grafted. That shows the accumulation of propane alkaloid. When on the stalk of datura scion of tomato is grafted, it shows accumulation of propane alkaloid. There will be accumulation of propane alkaloid will occur. On the contrary, when datura scions are grafted on tomato stalk, now the tomato stalk means major portion of part of plant is tomato, and in that datura scion is grafted, the production of propane alkaloid does not occur as usual. As usual, the concentration or amount of propane alkaloid that is accumulated on the datura stalk on which the tomato scion is grafted, that amount of propane alkaloid will not occur when the datura scion is grafted on tomato stalk, and show only traces of these alkaloid. Only small amount of or small traces of alkaloids are found. The above experiment suggests the possibility that the major site for biogenesis for propane alkaloid is root and no other organ of datura. So the propane alkaloid which is biosynthesized in datura, the major site for biogenesis of that propane alkaloid is root. Root is the major organ of biosynthesis of that propane alkaloid, and no other organ of datura they will synthesize the propane alkaloid. So from that we can tell that this is the site for biogenesis of propane alkaloid in that particular plant. So elucidation of particular biosynthetic pathway, that investigation is done by this grafting method, and what is the major site of the biogenesis of that particular secondary metabolites, that can also be determined by using this grafting technique. So these two techniques are used for investigation of biogenesis of secondary metabolites. If you like my video, please press the like button and subscribe my channel. Thank you.